Yeah, I will just a second. Right. Okay. Uh, Welcome everyone to the last problem solving session of the course Cell Biology. Uh, can you see my screen? Okay. Yeah. Right. So this is the last problem solving session of cell biology course offered by NPTEL. And the seventh session, good evening, thank you. The seventh session, which we could not do last Saturday, if someone did not join on the sorry, last Monday, we did it on Saturday, and like always, the recording and this coursework material PPT is available at the link. Okay. So today we'll finish off the course with the remaining set of topics that we taught to you in the last week of the course. And before ending, I'll just give you a mind map of what all we did. And hopefully, if you have any doubt, do use the chat box to reach out to me because this would be the last session. I have not taken any extra session for this course. So if you have any doubt or anything you want to clarify, you can use the chat box. Okay, so let's begin with the session. So this, I mean, today we will be discussing about the week 8 lecture notes. Week 8 had topics on special cell types. So we moved from plant cells, stem cells, nerve cells being neurons, and cancer cell, right? So we are basically talking about what are these specialized cells, cell type? Why are they special? What is the special feature about them? Because we, of course, are like aware there are so many different kind of cells, and we have read about their processes in general. So we have picked out the special cells which have unique features. Okay, so let's begin with plant cells, which is, I'm sure every one of us have time to time studied. So let me just give you a brief overview of what exactly in this course or what is exactly covered in the lecture notes, right? So plant, as we know, they show great diversity, but you see them different in the shape, size, structure, color, pattern, so many different things, right? Leaves can be different in color. The arrangement of flowers can be different. The flowers themselves are so different in variety and so beautiful. But all these plants, the seed plants especially, they follow a common structural plan, right? We know that we broadly classify the plant organs in three types: root, stem, and leaf, right? So you have root, which is there for the anchorage and absorption of water and mineral. You have the stem, which is there for the structural support, and the leaf, which is the most important uh, role of leaf is photosynthesis, right? And there are some additional terms which you would often come across, like nodes, which I just mentioned, and internode. Nodes is basically where your leaf is attached to the stem. Internode is the region between the nodes, right? Considering the fact that we are focusing more on the cell biology, I did not put those images, but I hope you can imagine where they are, right? Now, briefly about growth cell, because that's where we'll be zoom into cells. So generally, plant growth happens in the meristematic regions, right? They have the active cell division. You remember that plants have meristems, and meristems are the region where cell division happen. That too, mostly in the apical meristem. Apical, as a name suggests, apex, so they are on the tip of roots and shoots. That's where most of the bulk of growth, cell division happens. We also have axillary meristem, which is basically the lateral bud, which are for branch formation. So you have your roots and shoots, and then you have these branches, which also form, which also initiate from the meristem regions, right? Now we come to plant cell walls, because I said as we move into cell division, let's now connect the dot and let's go to cells and cell division. First of all, cell in the anatomy of the cell, right? So you can see here that the plant cells have cell wall, then a cell membrane, and then you have this cytoplasm, which has again the same uh, organelles that we have read previously also, for example, endoplasmic reticulum, nucleus, nucleolus, ribosome, peroxisome, Golgi apparatus, and mitochondria. But you have some organelles which are present only in the plant cell. For example, vacuole. Vacuole covers around 60 to 70% of the cell mass or cell body. So you have vacuole, you have amyloplast, chloroplast, and leucoplast, three type of plastids in plant cells. So if you go dig deeper into the plant cell wall, you will see, which let me just show you here once again. This outermost layer is the cell wall that we are talking about. This is not there in the animal cells, right? So the cell wall, which is in contact with the ECM, that basically has three layers. One is primary cell wall, then is secondary cell wall, and in the center you have the middle lamella. Okay, so what is this primary cell wall? Primary cell wall is basically the thin and the flexible cell wall that characteristic. It's present in growing cells and it is composed of cellulose, hemicellulose, and pectin. So it gives the uh, flexibility to the cell wall. Okay, and then the secondary cell wall, that is what makes the plant cells rigid and give it the strength, because these contain cellulose, and along with cellulose you have lignin. So in the secondary cell wall that makes the cell so rigid and with turgor pressure, that's because of the presence of lignin in the secondary cell wall. And at the center, which is middle lamella, you have calcium and magnesium peptide. Okay, this is basically, sorry, I meant outside. So middle lamella, which is a glue between adjacent cells. Okay, now along with this, you also see this plasmodesmata between adjacent cells, right? So what is this plasmodesma or plasmodesmata? These are cytoplasmic channels which connect the neighboring cells, and you can see there are two type of pathways to transport the nutrients and other water also, so other requirements. So you have this apoplastic and you have this symplastic, right? So if you see the apoplastic, you can see you just follow the yellow lines. They will only be through the cell wall. So I told you, right? The cell wall has middle lamella, which helps it connect. So you can imagine another such cell with a connection here. So if it's moving only in the cell wall via the cell wall, then it is an apoplastic pathway. And if any of the nutrients which moves from the cell wall via these connections in between, so the plasmodesmata will have a connection like this. Okay, in center to another center of the cell to another cell. So this will be symplastic. So these are there to efficiently meet the requirement of the plants, not only in terms of the molecules but also in terms of signals, also in terms of nutrients. Okay, so these are important features that you should remember. Now after this, in the lecture note, categorically, they picked up all the different nucleus. So they picked up mitochondria, they picked up amyloplast, leucoplast. So I have only retained the ones that are most important for in the pertinence of our discussion today. But it doesn't mean that the others are not that important, or I haven't got time to discuss them. I picked up those that were relevant to today's discussion, right? So the one of the most important new organelle in plant cell is this organelle here, which is called the chloroplast. And you can look at the structure here, right? The chloroplast name comes from presence of chlorophyll. Chlorophyll is basically a green color pigment. So that's why you will see often that chloroplast, and that also renders the green color to the leaves, right? And you will see inside the chloroplast these kind of structures. For example, first of all, it's also double membrane structure. So you have outer membrane, inner membrane. And then when you look inside the inner membrane, you have these granas. Granas are basically stacks of, so you can imagine coins, stacks of coins. So this is one stack, two stack, three stacks. So this is one grana, two, three, four. Now inside the grana, if you see, these are, the grana are formed of thylakoid. So each of these is called a thylakoid, right? And then inside the thylakoid, you will see there is lumen. And whatever is not grana here are the stroma. Stroma is basically the cytoplasm as we call in the cell. We call that remaining part in the chloroplast as stroma. Okay. Now, what is the importance of chloroplast? As I said, these are plastids. There are three types of plastids in plant cells: chloroplast, amyloplast, leucoplast. All have their own features. Right now, chloroplast. So there is also, along with the two membranes here. Sorry. Uh, I don't know why is my cursor not working. Sorry, I have to. Is my screen moving? No, right? Okay, doesn't matter. I will just reshare it. So you have these two layers, outer membrane and inner membrane. In thylakoid also has its own membrane. That's why you have the lumen. So there are three kind of membranes that you see in plants: chloroplast, outer, inner, and thylakoid. So in the thylakoids, you have the chlorophyll and carotenoids, and they carry out photosynthesis. Photosynthesis is basically the mechanism by which plants convert the light energy into chemical energy. So you see that here. So they fix the carbon dioxide, use up the water in the presence of sunlight and chlorophyll, and make it convert it to glucose and oxygen. Oxygen is released back to the environment. Glucose is used up by the plant cell. Now this is uh like the understanding is similar from glycolysis, where our body uses up glycolysis for our own cell processes. Here the oxygen is released and glucose is used up, is made for again the same purpose of cell requirement, cell processes, right? The only thing is here we are uh, utilizing the carbon dioxide and the water, and that is this carbon dioxide it is taken in, which is a carbon containing gas. The carbon from here is then assimilated, and then this glucose is formed. Okay, now not going into the details of this, just an overview. That's why I gave the equation there. And you should remember that photosynthesis have takes place in two stages. There is light reaction and dark reaction. The light reaction basically that part which talks about the usage of light, which will be here, how the light is used and how is chlorophyll part of it. Basically, what I'm trying to allude to is when you look at chlorophyll, you will see that it's pigment, and then at certain threshold wavelength, it will capture the sunlight, and then how that is translate, like how the electron is transferred till it is forming glucose. So basically, that's what I'm trying to tell, that there is a lot of reactions here, and the ones that involve light are called the light reactions, and they take part in thylakoid membrane, and the outcome of light reaction will be ATP and NADPH. Okay, then comes the dark reaction. In this, the light is no more needed because you've already taken in the carbon dioxide. That carbon dioxide is fixed to form the carbohydrates, and this takes place in the stroma. So like you can see here, this is stroma here. So you have the dark reactions take place here and the light reactions take place here. Okay, now so this is chlorophyll and photosynthesis. The next topic that was discussed was plant mitosis. So why is it? Why did this specifically discuss mitosis? So it is saying it's otherwise the stages are same as that of the animal cell that we have read about. The unique features are of plant cell mitosis is that they have the pre-prophase band. First thing, okay. Why pre-prophase band? Here you can see, right, these bands are formed in the pre-prophase. That's because they have to make this cell wall. It's not just dysfunction of two cells, they also have to make cell wall, so their division plane has to be correct, right? So you have a pre-prophase band which is unique to plant cell. Second, they don't have centrosomes that we have been discussing all throughout in the animal cell, and we discussed exclusively about the centrioles, MTOC, all of that. That is not present in plants. In that, in place of that, the spindle fiber which is formed in plant cells is because of diffuse microtubule foci. So the foci is a location where the microtubules are arranged and how they form at the axial ends. They don't have MTOC or the like the centrosome, which actually begin the formation of microtubules. So there are some small differences that you should remember. Okay. Now, the next thing is these hormones, which also I think everyone is almost aware of. So there are these plant hormones are basically the signals link components. They give signals, and respective changes happen in the cells or in the physiology. For example, here you have auxin. So there are, so let me just go here. Okay. So you have five types of hormones. And that's why I did not say they always promote growth. They elicit uh, the response that is amenable or that is dedicated for these compounds. For example, here the hormones can be of two types. One, promoting the growth of the plant cells. the other inhibiting the growth of the plant cells. So the first one which promotes are auxin, cytokine, and gibberellins, and the inhibitors are the ethylene and abscisic acid, and they have their very specific roles to play in plant metabolism and physiology. For example, auxin, which is responsible for growth, is found in stem elongation and apical dominance. So all the apex growth is because of auxin. Gibberellin is required for stem growth. Okay. Cytokinin, the name tells what role it can play. It is for cytokinesis, so cell division. And the ones that stop the growth, why would you want to, why would you want to prevent the growth of cell? So in case of abscisic acid, that's because you want the seed to undergo dormancy. So if there is any stress in the environment, these hormones are released, just like how in animals also, right? Us also, we have the release of epinephrine, non-epinephrine in response to our surrounding. Similarly here, in case there is stress, you have abscisic acid that leads to seed dormancy and ethylene that leads to fruit ripening, right? So if you want to dedicate your time to ripening your fruit, and then that requires next level of regulation, then your focus has to move towards the fruits and not on the cell division. So the cell division is stopped for time being, and the focus shifts to the fruits, right? So that's why you have ethylene that is doing that job. Now, another last topic that was discussed in plant cell before I finished for plants today is auxin inducible Dragon system, which is a very unique and a very elegant system. So basically this system, as you can see from the slides here also. So what does this do? Degron is basically any sequence that leads to protein degradation. So people have now used up this hormone and the way the proteins respond to this hormone to basically use it for catering or doing minute changes in the system. For example, let me just explain this entire procedure to you. So this AID is basically a conditional protein degradation technique, conditional being that it is under the control. So it basically we can target protein inside the cell and we can do it in an inducible system. Inducible being this is under the control of auxin. Okay. And what principle does it use or use ideas from? The auxins, all of these hormones have their own signaling pathway because of which they reach to the their target location. So in this system here, we use the auxin signaling pathway, and then we basically change the protein target for the targeted destruction. Okay. So here the auxins or cytokines, they are broad classes. You have compounds in these which are belonging to this class. So one of these classes or one of the proteins or one of the components which are part of the auxin is indole 3 acetic, also called IAA, and that is what you're seeing here in the slide. IAA, auxin. Okay. So now what do? You use this auxin hormone IAA and you promote the degradation of certain proteins by using the ubiquitin proteasome pathway that we have already discussed in the previous sessions, right? So what do they do? For the protein of interest to be the target of the system, you add an AID tag to the protein of interest, okay? And then you also go to the TIR, which is basically the receptor of this protein, and you use that to interact with your auxin, right? So you have an AID tag. That tag is degron. The name I told you last time also, the name of the proteins generally come from their activity. So degron, degrading. So basically they are degrading a particular protein. So you add that degron tag onto your target protein. Then you have the receptor, which is called TIR in this case. Let me try to show you here. Right, TIR. So this TIR is called transport inhibitor response 1 protein. Now this is a receptor that forms part of the ubiquitin ligase complex. Okay. And then you have auxin, which is basically binding to the cells. So what you do first thing in this protocol is that you make this AID degron tag and you also express a TIR. Okay. Then in general, without adding auxin, if you don't add auxin, this will just remain in the cell. The moment you add auxin, what happens? Auxin promotes binding of TIR to the AID tag. Why? Because TIR the receptor which binds to auxin can then bind to the AID tag. So it binds to the AID tag, and as soon as it binds to the AID tag, it because it also has domains for SCF, which is your ubiquitin complex. So it brings ubiquitin to the target protein as the ubiquitin tag, and then brings the proteasome and degrades. So you have very creatively engineered the natural signaling pathway to cater to experimental needs. So that's what is happening here. The moment you have a protein in interest and you have your auxin binds and forms this entire complex, which leads to the proteasome mediated degradation of the protein of interest, right? Okay, so let's do some questions on these topics so that it becomes easier for you. I've tried to keep it as minimal as possible because there are a lot of topics that had to be otherwise covered, and we have also 5 lectures to cover today. So let's just keep it going. Okay. So yeah, the first set of questions is from the plant cells are this. Plasmodesmata in plant cells function to do what? What is the role of plasmodesmata? Then you have plant cells, uh, cytokinesis. How does cytokinesis or how does the breaking of the cell happen or division or formation of two new cells happen? The middle lamella of plant cell walls, what is it composed of? And the plant cell wall is flexible and found in growing cells. Where in which layer do you found the growing cell, which we have discussed already? Okay. Anyone else? I'll wait for 1 more minute. Okay. Right. Let me go to the answers here. Right. So plasmodesmata, as we had discussed already, it is connecting these cytoplasm of adjacent cells. In plant cells, cytokinesis occurs because of cell plate, and that is what we told when the pre-prophase band I was discussing. I told that cell plate is what is required, what is required for cytokinesis. Middle lamella has calcium pectate. We already discussed that, and the flexible part of the cell wall is primary wall. The harder part is the secondary wall. Right. Next one, secondary cell wall, which is why is it hardened basically? What is it composed of? Which organelle is crucial for photosynthesis? Now, which of these is not a plant hormone? And very simple question, right? What does it convert CO2 and water into for photosynthesis? Yes, this is the last session because this is an 8-week course. So we have 8 problem solving. This is the 8th problem solving session. So this would be the last one. B triple C. Okay. Anyone else? I thought the PPTs are already available. The link is given to you where the PPTs are available, right? In fact, I think I shared the link here in the chat box as well. Let me find it out. The PPTs and the recordings of the lecture videos are always uploaded the very same day, but I will just share the link again. So I've sent the link for the Google Drive. Let me know if you can access the because I thought that was being done already. Thank you. It's okay. Welcome. No problem. Okay. So let's move to the next question set. So secondary cell wall contains large amount of lignin, starch, and lipid. We already know that. So lignin is the correct answer. Which organelle is for photosynthesis? Chloroplast. Now, which of these is not a plant hormone? Insulin, right? That's not a hormone, right? Plant hormone in that perspective. And photosynthesis leads to the formation of carbohydrates. Okay, next one. So in the auxin signaling pathway, the presence of auxin leads to which molecular event? Second, which of the plant hormones is responsible or directly associated with food ripening? Which plant hormone is primarily responsible for promoting stem elongation and also inhibiting lateral bud growth? And which feature distinguishes mitotic spindle organization in plants from that in animal cells? DDBB. Okay, anyone else? CDAB. All right. Okay. So it's C, D, A, and B. Let me try to walk you through. So the auxin signaling pathway, like I said, if you have auxin, it leads to the polyubiquitination and degradation of auxin repressor proteins, right? So the first option is inhibition of SCF activity. That's exactly what we want. We don't want it to get inhibited. Stabilization of transcriptional repression. That is not the correct one here. We want the SCF and the receptors and everything to work. So we don't want repressor to be there. That's why we have to remove the repressor proteins. Auxin does that. Auxin removes the repressor proteins. Okay. So you ubiquitinate and degrade that. And the next one was direct activation of transcription factors by auxin binding DNA. No, that's because you need this degradation of the repressor protein, and for degradation is why you need this your entire auxin, TIR, STF coming together. For fruit ripening, it is ethylene. Okay, abscisic acid was for the other one, the contact ones. And then you have, okay. So primarily responsible for stem elongation. Any elongation is because of auxin. And because I told you they have this apical elongation, when so auxins promote apical elongation and inhibit lateral elongation. Okay. And what differentiates plant from animal cells in terms of the mitotic spindle? There is no centrosome, which I told, and you have this foci, localized foci, which move to the opposite pole and they form the microtubule spindle fibers, right? Okay. So I hope that was somewhat clear, because there is a lot of thing to cover in our plant cell, and it is very unfair to, for like just 15 minutes or 20 minutes won't cover it. But I hope that you got the gist of the lecture. What are the different topics that they tried to explain? Okay, the next specialized cells were stem cells, which we are fairly aware of what these are. So basically, as we all know, stem cells are those cells that can get renewed, that have capability of renewal, and they can enter cell cycle. So we will see all the features that make it a stem cell, right? So now, before stem cells, to understand stem cells, you should also understand that in general, you have some cells, so first cells divide rapidly and then they differentiate into special types. So all the type of cells that you see in the body, neurons or muscle cells or fibroblasts, any cells of these sort, all of them have divided and then differentiated. When you differentiate is when you become a particular type of cell, right? So I've differentiated. Now, these differentiated tissues or differentiated cells are the ones that form tissues and then form the organs, right? That's the hierarchy, or that's the classification that we have read in the cellular organization. You have cell, you have tissue, you have organ system, and you have organism, right? Now, many of these differentiated cells that have become a certain type, they exit the cell cycle because they don't have to undergo cell division anymore, and they enter this permanent cell cycle arrest stage, which is called G naught, which we have seen in neurons. Okay. Now, that is a case, right? Now, also you have sometimes cells getting damaged in case of injury or proteasome degradation method facilitated. So you have some cell dying, you have some cell getting damaged. Now, these cells require replacement, right? Because in a tissue, you have very proper organization, and if some cells die, you need to replenish it. So there are also some differentiated cells that can re-enter the cell cycle. The ones that do not enter are, for example, neurons, but there are ones that can enter also, even though they are differentiated. For example, fibroblasts. These are basically involved in wound repair. So these have their own growth factors. So they have their own signal. For example, PDGF, here, platelet-derived growth factor. So the moment there is platelet because of wounds, you have fibroblasts coming and filling in that patch, right? That's there. Then you also have endothelial cells, which are on the lining of blood vessels, which we also looked in the previous session under the signal from VEGF, vascular endothelial growth factor, which it leads to the formation of new blood vessels, right? Angiogenesis. And then you also have organs which are capable of regeneration, for example, liver cells, right? So liver cells can regenerate them, right? Now, how can these regenerate? How are they able to become differentiated? And then upon loss, they are able to come back to the same differentiated form. That comes from the idea of stem cells, right? So these stem cells are self-renewing cells. They can divide repeatedly, and they can provide, once they are, so they can provide the cell of stock of new cells. And when they are producing, so one of the cell becomes stem cell and one of them become differentiating cell. So the stem cell still retains the information for the next time. The differentiated cell has formed the desired cell that the body wants, and it has got deposited the required region. But the one that has formed a stem cell is available for any further round of cell division, right? So you have these stem cells which are basically able to form different kinds of cell that you can see around, right? Now you see here in the slide, these are the regions where you can find stem cells, which can also regenerate. For example, red blood cell, I just told you. You also have stem cells in fat cells, cardiac muscles, enterocytes, kidney cells, chondrocytes, epithelial cells. You also have them in neurons. and the most important is from blastocyst, which is basically when your uh, whole cell body is, or your zygote, in your zygote you have this blastocyst. It makes sense, right? Because these are the cell stalks, and from zygote you have the entire body plan and cell structure coming up. So it is definitely present in the blastocyst, right? Now, uh, next feature about stem cell is these stem cells, as you can see from the names also, they are very different environment, right? So stem cells have their own niches, have their specialized microenvironments. The blastocyst cells, stem cells have a different environment there, different factors, different hormones, different kind of surrounding nutrients. While the one which is in red blood cell will have a different kind of cell, different kind of environment. So stem cells, they exist in their specialized microenvironment, and these are called niches. And because they are present in this I'm talking about this part here. So because they have their own niches, they have you know, the cells from that particular stem cell can become differentiated into a particular type of cell, and they have their renewal because they have soluble factors, and we also have maintenance because of the, even after division you have a stem cell forming and the differentiated cells, right? So you would often find these stem cells in the tissues, in the organs, which are undergoing continuous replenishments, right? So you can have, like I said, blood vessels, epithelial, skin epithelial, you have continuous erosion or falling of skin. So skin epithelial cell, intestinal, sperm cells. So you have, but then you don't have tissues or such stem cells in some parts of the body, and that was mentioned as in the lecture note, being the retinal epithelium and auditory, so the sense organs. So we would imagine that if cells can renew themselves, such organs, if they get any problem comes to these organs, they can also be corrected. But that's not the case because of the factor that in retinal epithelium or the auditory epithelium, you don't have stem cells. So that's why once you lose your sensation of, or once your eyesight or once your hearing signal is lost, you cannot, basically go back to it again, right? Okay, now, so I hope you understood that stem cells are basically not only important for proliferation, they are important for differentiation, for migration, and for the survival, because of the fact that there is a cell stock that is maintained, right? Now, before I move to the other topics of stem cells, there are some concepts of stem cells that you should definitely remember. One being how they are classified, right? So you can see here, there are stem cells, can be embryonic stem cells, fetal stem cells, and adult stem cells. Embryonic when you are the zygote, fetal when you have got implanted and the three stages of implantation has taken place, so morula, gastrula, blastula. And then you have the adult stem cells when your cells have started becoming differentiated. At each of these cells, the type of stem cells and their potency difference, I explained to you. So you have totipotent, pluripotent, multipotent, oligo, and unipotent. As a name suggests, these are the different kind of cells that these stem cells can give rise to, right? So the first cell here is totipotent here, right? Which comes from the embryonic stem cells. Toti, as the name suggests, it can give rise to all cell types, right? Including embryonics and extra-embryonic. So these are the cells that have the capability to differentiate into different and any kind of cells, right? You find them in zygote and you find them in the early blastomeres. Why? Because you need to create the entire organism from them. So these can give rise to different kinds of cells, right? So toti. But the moment it loses this feature of forming all different type of cells, the next level is pluripotent stem cells, right? Which is here, which also you get from embryonic stem cells. The only difference is that pluripotent stem cells can basically form all the body cell types, right? So you can have all the germ layers or anything that is to do with embryonic cells, except you cannot form the placenta. Totipotent could form the placenta also. Pluri, so you can see, you can divide it like this, total being everything. So total, pluri means multiple. Okay, you can make a hierarchy in your mind how they can form the different components. Now here you have embryonic stem cells as an example, and you also have iPSC, if you remember from the lecture notes, induced pluripotent stem cells. We will explain and discuss this in coming slides also. Okay, now pluri is plural or multiple, but you can also give, probably you can put it as plural type of cells, because the next one is again multipotent, also forming multiple different cell kinds. This also comes from fetal stem cells, right? So what is multipotent? What is the difference between pluri and multi? Now, multi is basically those stem cells that can produce multiple cell types, of course, but within a specified lineage. Okay, so when you give the name to the stem cells, say hematopoietic stem cells or mesenchymal stem cells, these stem cells can then only give rise to cells which are for that particular lineage. Hematopoietic can give rise to any blood cell. There are different kind of blood cell that we will see in the next slide. So one cell, one stem cell can give rise to all these different kind of blood cells, but only blood cells. When it does only blood cell, it becomes multi. So you can imagine multipotent to be a particular lineage's multiple cells. Pluri means still different kinds of cells. Okay. Right, for example, and then you have mesenchymal, which is for the fats and cartilage, which I showed in the previous slide. Okay, then you have oligo and uni. Uni, as the name suggests, that it can give rise to only one cell type, right? But it can still retain the ability for self-renewal. So it can basically form another cell, and but still retain the ability for self-renewal. And oligo is basically it can give rise to 2 or 3. Definitely not as many as multipotent, but definitely higher than unipotent. So there is a gradation in what kind of cells they can form and which type of cells they can form. So that is important for you to remember. Okay, now, like I said, an example of the yeah, multipotent cells. So I told you two examples, hematopoietic and mesenchymal. One of the example I'm showing here, hematopoietic is in bone marrow. You have hematopoietic stem cells giving rise to two types. First, myeloid progenitor cell, lymphoid progenitor cells. Then you have myeloid giving rise to 6 different types here. Megakaryocyte, eosinophil, basophil, erythrocyte, monocyte, neutrophil, and these can again give rise to dendritic cell and macrophage and the platelet. And then on the other side, the ones which are associated with lymph, you have T, B, and natural killer cells. B cells can also give rise to memory cells called plasma cell. All these different kind of cells at different steps that you saw can come from one single stem cell, right? The only thing is which lineage it takes in the next commitment. Does it go for myeloid progenitor or does it become a lymphoid? It determines which kind of cell. But in both the methods, you have multiple different cell kinds forming, but all of them are under the singular term blood cells. So when you only form for a particular lineage, you are called multipotent stem cells, right? I hope that is clear. Okay, now I just wanted to give an example of this. Now let's come to this iPSC, which is very, very important and interesting also. So first of all, let me explain to you what is iPSC. So iPSC is basically induced pluripotent stem cells. The name suggests, right, that you have done something, you have induced something, and then you have made it pluripotent in its nature, right? So what are these cells? These are basically pluripotent cells that have been generated by reprogramming differentiated adult cells. So remember, when you are differentiated, you have lost the capability of becoming these different kinds of cells because you have become a erythrocyte, you have become a basophil. So how do you go back to form? So let me just take you back one step. You have already become basophil. What makes you go back to progenitor? That's not the normal way, right? You have become basophil from MPC, from this myeloid progenitor cell. So then what makes this basophil go back to the stem cell state condition? So that's when you have these important features or important concept here, which is basically induced pluripotency, right? Now, once you reprogram these differentiated cells back into the pluripotent stage, these new cells which are pluripotent in nature behave the same way the other pluripotent nature, which are embryonic stem cells, do, right? Now, how do you do this? How do you reprogram these cells? So these were, as you can see on the slide also, right? These are, so you can see there is hematopoietic stem cells, you have epithelial cells, all these cells which are forming. You can make them induced pluripotent cells and you can basically revert a step back. Okay. Now, these were first generated by Shinya Yamanaka, who also received the Nobel Prize for this. I'm sorry. Right. So, how do you do these iPSCs? What are the factors that bring it back to the stem cell state. These are, as also it is seen on the slide here, OCT4, SOX2, KLM4, and c-MYC. Okay, so you can give your own mnemonic to remember that. You can basically SOKM, you can remember it like this. So always try to make a mnemonic so that it becomes easier for you to understand. Okay, now what he did, or what you generally do in reprogramming, you have these adult somatic cells, differentiated cells, right? For example, fibroblast here. Then you give this Yamanaka factors. These are this collectively are called the Yamanaka factors, and you introduce these factors via different vectors. Now, once these factors have reached the cell, they reset the gene expression program of the cells, right? And these factors can then take the cell back to the pluripotency stage, and that's why you then take this iPSC colony and use it for your growth, right? So you are basically mm, yeah, you have your iPSCs, and then you take these iPSCs and you give other differentiating factors and you make them into different kind of cell, those type of cell. So just imagine the idea here is, in very vague terminology, you have a fibroblast and you want to make a hepatocyte, right? So if you take it one step back and then make it fibroblast, sorry, hepatocyte. So you can make, very broadly speaking, you can make one cell type into other cell type just by reprogramming it. And that can be done because of these Yamanaka factors, right? At the same time, not only making them, you can also retain these iPSCs as pluripotency stage, self-renewal, and also make it according to patient specific. That's the application actually. So why do we actually do this? We do this because this is a part of regenerative medicine, tissue repair. So you can make particular type of cells which require, which are important or missing from the tissue. Similarly, cell replacement here, therapy, you have some cells which are missing, for example, neurons and got degraded. So you make cells as neurons or cardiomyocytes, something like this, right? Regenerative medicine, a very upcoming field. Then you have disease modeling also. So you can, so the same cells, for example, we have diseases which affect particular cell types, right? Brain cells or nerve cells, or diseases like Parkinson's, Alzheimer's. You make your own cell type and then study the features in that cell type without the in need of any other external factors. So you also do it for disease modeling. You try to understand how these diseases progress by making your iPSs. You can have drug screening, the same idea that we have a cells in our hand, we can test everything that is required to be on the cell at in vitro level, right? Okay, and why do we want to do this on in vitro? Because uh, generally, if you want to do for stem cells, you would need it from embryonic stem cells, and that's very problematic, or sometimes it comes under huge ethical issues, right? So you generally avoid that. Uh, Sometimes you can, or because you are taking cell from a patient and then you are regenerating these iPSCs, you are basically catering the treatment according to the patient. So it's a very patient specific cell therapy. So there are very less chances of it being rejected. And you can, if it's in vitro, you can, repeatability is very high, so you can do it at a greater scale, right? So that's always there. Right. Now, so I hope you understood this part. Now we will talk about something very interesting, which is Allen cell collection. I hope you remember from the lecture notes. So the lecture began with the term called Google Map of Cell, which is basically, if you know Google Map, we all know Google Map, right? It basically tells you the direction of how to move. So something similar for cell, how do cells are organized, where, what is organized, what is kept where. So something like this. So for that, they have this uh, the collection here, which is called the Allen cell collection. So let me explain to you. This is the concept taken from iPSCs. So it makes sense to read it here, right? So these are genome edited iPSCs from human. Okay. Now, this was developed by Allen Institute for Cell Science. That's why the name Allen Cell Collection. And the idea here is to study the cell organization and the dynamics, not only how they are organized, but how they are interacting with each other, what protein is moving where. And you do that mapping using a fluorescent tag on the proteins, right? Now here, the idea is that, uh, so otherwise, if you have to do such dynamical study, you have to have fixed cells that you have fixed. Then you have put antibody staining. You have your protein, you have antibody staining, and you track the cells and static images, right? So what does Allen cell do? There are three, four techniques that I have integrated here. One is live cell imaging that makes it so beautiful to even look at. Right. Second is endogenous protein tagging. So you basically are dealing with cells or proteins inside the like proteins inside the cell. And then you have microscopy, which is 3D and time lapse. So you can capture not only for a particular time window, but you can look it across their entire distribution. So it's very elegant also, right? Now let me try to, so the goal here is to make a systemic map of the cellular structures, which you can see from the slide looks just beautiful. What kind of cells are present where, how they are present, and which lining they are present. It's just amazing to see this, right? Now, let's try to understand what are they trying to do? What are the mechanism, right? Now, I told you about few terminologies here. They used fluorescent protein, they use live imaging. So let me just try to put it all together. So the steps here is first you have to select your protein here. So any protein, tubulin, actin, mitochondrial proteins, anything. You edit your protein to put in a GFP. And that is very much doable in today's age because you have CRISPR-Cas, you have gene editing. Earlier it was viral mediated, retroviral vector mediated, so many stuff. But now you have an elegant method called CRISPR-Cas. So you can enter or insert this particular tag into your genome or your protein. Now express the protein, which will carry this green signal inside because it's a green protein, green fluorescence protein. Now you have the cells expressing the protein, which is up with a tag. Now you image it uh, under your microscope. Very easy because it's a fluorescent fluorescent protein. So use a fluorescent microscope. Look at these green colors and see where they are. And don't just stop at microscopy. Don't just stop at looking it. You can also image it as time lapse window, right? So you do that. Now, where are these coming from? What is the importance of iPSC here? So you can have the iPSC and you can see where these fibroblasts grow and bind or grow and stay. Where this particular type of, so sometimes you need to tag the protein, you need to know the protein, you need to know the sequence. But if you have iPSC, you can control the differentiation of the particular cell and make it a particular type of cell and then regulate it at the genomic level because at in vitro condition, your CRISPR-Cas is very efficient. You can control and manipulate those. So you have iPSCs, which are the beginning source material. So you don't have to even worry which cells to get from and where to get from. So that's the idea. That's how iPSCs are connected here. So even in general, also you first culture the iPSC and then you put in the genes of requirement because you whichever cells. For example, here I showed you image of this is anybody idea what is this? This is brain map, right? So I know which cells I don't want to have in my design. So I basically, when I use iPSC, I give certain factors which make it certain kind of cells which are found in brain, tag them, edit the genome using CRISPR-Cas, put the GFP, and then just visualize where they are, right? Now you will say why there are different colors. GFP is just one of the fluorescent protein. You can put multiple tags to give different colors. You can do different kind of staining. All those are there for sure, right? So the idea, as you can see here from this image on your slide, you'll get to know the cell organization, you'll get to know dynamics, where do they move, how do they move, and this is very applicable in terms of disease and drug discovery, right? So that's the whole idea about your Allen's cell culture collection or the idea of what drives this whole Google map of cell, right? Okay, so that brings to an end to this part or lecture, second lecture of the week. Let's do some questions so that we get idea about the topics more. So the first thing is stem cells are characterized by which of these factors? What are embryonic stem cells derived from? Where are they derived from? If you have stem cell, what can they form? And totipotent stem cells can form which kind of differentiated cells? Easy questions. These are all simple features of stem cells. Anyone else wants to take a guess? I will wait for 1 minute more. Okay. B A N A. All right. Anyone else? Okay. So I'll give the answer. Just a second, sorry. So B, A, and B. I hope that were the answers. Mostly that, right? Yes. So the first one here says stem cells are characterized by self-renewal. These we have already established, right? That stem cells can have two defining properties: self-renewal and potency of getting differentiated into different type of cells. Now, the other reasons here, or the other options, limited division, apoptosis, and this is are the very thing that stem cells don't do, right? So stem cells don't have limited division; they can divide many times, right? What is for which cells it is differentiate, for which cells it is basically limited? It's the differentiated cells, right? That's why they have problem. Apoptosis, uh, it's not a feature of stem cells, which is basically programmed cell death, and senescence, it doesn't enter cell cycle arrest. Next one, where are the embryonic stem cells derived from? Blastocyst stage embryos, as the name also suggests, right? Not from the adult tissue. That's not the same as embryonic stem cells, not umbilical cord. These are umbilical cord blood. Blood means hematopoietic stem cells, and bone marrow are again hematopoietic stem cells, right? So both of these cord and bone marrow are hematopoietic. So the embryonic stem cells come from the blastocyst, that too from another term that I didn't mention, ICM, inner cell mass, okay, that you will often encounter. Next one, stem cells differentiate into specialized cells that we have already established, right? Only muscle, only neuron, only blood vessels, those are not the correct terminology. And lastly, totipotent, toti means totality, so entire organism. Okay, let's move to the next one. So next is pluripotent stem cells. Which of these can be the cells from such kind of stem cells? Adult stem cells are also called what type of stem cells? Stem cell therapy. Okay, what do you use it for? We have discussed this already. It's just another term for the application. And what the stem cell maintain the tissue. How, so they, I told you that they are also responsible for maintenance of the cell. What is the, why is that the reason? Triple A, B. Okay, let's look at the answers. A, and B. Right, that's great. Uh, so the first question was pluripotent. So this pluri means multiple or many different kinds of cells. So these are again embryonic stem cells or iPSCs. These can differentiate into almost all cells, not as much as totipotent because these are mainly for the embryonic cells, not the placenta, right? So ectoderm, mesoderm, and endoderm. Adult stem cells are called somatic stem cells. That's because they are found in body tissues, which are called somatic tissues. So somatic stem cells. Stem cell therapy for regenerative medicine, we already discussed that. And stem cell maintains tissue also because of self-renewal and differentiation. They basically generate the specialized type of cells that are required for tissue repair and function, right? Okay. And the next set. So hematopoietic stem cells will produce which kind of cells? Induced pluripotent stem cells can be created by stem cell niche refers to what in terms of the stem cell features, and if there is loss of stem cell regulation, what can it lead to? Easy questions, very easy. These are not very difficult. A, B, B, A. Okay, let's look at the answers. A, B, B, and A, right? You guys are getting it all correct at one go. That's great. So hematopoietic stem cells produce blood cells, as we already know of, and they can form different kinds of cells, red blood cells, white blood cells, platelets. Now, just for your information, muscle cells, the stem cells are called mesodermal progenitors. Okay. Skin cells, epidermal stem cells, and neurons. Neurons are neural stem cells, right? Okay. Now, induced pluripotent stem cells are created by reprogramming adult cells by using the Yamanaka factors, SOC4, SOX2, KLM4, and c-MYC. Remember these. Stem cell niche refers to the microenvironment which supports the stem cells because they are the specialist microenvironment where the stem cells reside, and they regulate the stem cell behavior by providing the cell-to-cell interactions of different kind of cells, the growth factors, the signaling molecules, and the ECM. So they are the niche, they are the habitat, you can say, for these stem cells. And if you have loss of stem cell regulation, it can lead to cancer. Why? Because these stem cells are able to proliferate. So if they proliferate and differentiate without control, they can lead to cancer. And this is one of the reasons, one of the primary cells from which cancers, generate or origin. Okay. Now, if you remember in the lecture note, this part of stem cell was covered in two parts, part one, part two. So the part one was mostly understanding stem cells, and the second lecture was most about applications. So we'll now do three more sets of questions for your application part, so that the topics are not missed out, or like you can understand them. So let's move to the next part, which is what is the primary goal of creating Google map of the cell? Which three main technologies were combined in this study to analyze cell organization, right? So this iron cell collection. Uh, what is Cas9 doing in CRISPR-Cas, and why are fluorescent tags introduced into gene of encoding structural proteins? Anyone? Just give it an attempt. Even if it's wrong, it's all right. That's the whole idea. These look like very next level, but that's what we're trying to do here. B, triple BC, anyone else? Okay. Okay, let's move to the answers because it is not that difficult. But yeah, I understand because we are doing it for the first time and that. Yeah. Yeah, you have IPSs because you make you reprogram the differentiated cells. So you use IPSs and then CRISPR-Cas9 for your gene editing. Right, and then CRISPR and Cas9, what do these refer to? So Cas9 is basically the one that cuts DNA at a specific target so that you can introduce your desired gene of interest, or yeah, your design gene of interest into the genome of the cell or the sequence of the cell. And the last one, why are fluorescent tech proteins introduced? It is to visualize cellular structure during live imaging, because otherwise you have to do other type of staining where you can localize those proteins and see, right? So it's better to do live imaging because you can also look by color. Next, which stem cell type was used as a model system in the study? Why is endogenous tagging preferred over traditional overexpression methods? What is the main purpose of this collection, and which cellular process was frequently observed using live? So which is the uh, process that was mainly observed? Anyone? Okay, I'll wait for a minute more and then we discuss the answers. Okay, so let's look at the answers. iPSCs we already know about. Why is endogenous tagging better than the traditional methods of very overexpress a particular protein? It's because yes. Okay. When you are tagging your protein inside the cell, you are ensuring that the protein is in its normal localization, normal conformation, normal expression levels. Instead, when you're overexpression, there could be artifacts like it is abnormally located, or it goes haywire, or there is something which also goes aberrant along with the overexpression. So there can be many problems which you can encounter if you do overexpression. So endogenously tagging the genes or the cells is a better option. And this we already know that ACC or LN cell collection is for creating a library or a collection of all these stem cells lines. And lastly, which one is the most used method? It is mitosis and cell dynamics, where what is the idea? You want to observe the microtubule behavior, nuclear envelope changes, and organelle dynamics, and all the different processes that we had discussed in cell cycle, right? How this DNA compacts, how does protein move from one end to the other, how does spindle fibers move, all of these different things can be studied by this method. Okay, and the last questions for this lecture. What challenges arises when analyzing these thousands of live cell images? Why do researchers compare gene expression with cellular organization? In cardiomyocyte differentiation studies, which structure was used to study muscle organization? And what is the long-term goal of integrating imaging, gene expression, and computational models? Anyone? Just take an attempt or just take a guess of what could be the answers. It can help you at like navigate your thoughts in the direction. Why do you think these advanced techniques are coming up even? Okay, CCBA. Okay. Anyone else wants to take an attempt? A minute more and then I tell you the answers. Okay, let me tell the answers. So B, B, C, and B. Okay, let me walk you through. So what challenges arises when analyzing thousands of live cell images? So the cells that we generally study, they vary in their structure, shape, size. So to get the proper information, it takes time, and that's the challenge, right? So you have difficulty in extracting and segmenting the structures from these images because not all of them are similar kind. So you have to take each case as it comes, right? Okay. Now, why do you want to compare gene expression with cellular organization? Because we want to understand how does genotype, which is when you change an expression level, or how uh, when a protein is getting expressed, how does that change the phenotype? How does it lead? How does it lead to the changes in the structure, organization. So we have to look at the genotype and the phenotype relation. That's why we start by comparing gene expression, right? If you're doing cardiomyocyte, you go for sarcomere, which is the unit, contractile unit in case of cardiac muscle cells. And what is the idea, long-term goal of this entire endeavor? It is to predict cell behavior and cell state transition. So all the cell state behavior that we are currently trying to understand, if you use you have images glossary and you use ML, which is AI or machine learning or any other such advanced technology, to so that you can see, currently you see this genotype, you have this phenotype, then you see an aberration here, you see this different kind of phenotype. So this information can be used in the future to predict what kind of behavior the cell can show depending on its genotype. So to get that kind of understanding uh, is where this will come handy. That's a long term idea. Okay, so let's move to the third lecture now. We'll quickly finish neurons. So we already are aware of neurons, right? We already know what's neuron, what they're doing, and why is it a specialized cell. So neurons are basically those cells which are responsible for communication in the nervous system, right? So there is a lot to cover in neurons, and I only have limited time. So I will just cover broadly what was discussed in the lectures, right? So broadly, neurons is an excitable cell. It is able to receive, process, and transmit information via electrical and chemical signal. If you look at the structure of neuron, which is on the slide here, you can see there is cell body, there is dendrite, axon, and myelin sheath, which you see here, and rods of Ranvier, right? Now, quickly, the cell body, also called soma, it contains the nucleus and the organelles, and dendrites are this extension which reach out to other neurons to receive the signals and carry the signals. Axon is basically carries the signals in the neurons. Okay, and myelin sheath is basically this insulation of lipid layer around the axon. And you have two type of cells here, squamous cells and oligodendrocytes present in respective nervous system. So you have peripheral nervous system and you have the central nervous system, right? So peripheral nervous system has Schwann cells making up the myelin sheath, and central nervous system has oligodendrocytes, right? And then you have these nodes of Ranvier, which are the gaps in the myelin sheath. Now, why is there these kind of sheathing? Because you are propagating impulse from here, and these are neurons, and these are connecting one part of the body to the other part, and you want it to be very quick. So these let you to do saltatory conduction or jumping. So the jump here comes here because it's insulated, it doesn't get conducted here, jumps to this part. From here, it jumps to this part. So you have these nodes of Ranvier, which allows the quick transmission of impulses, right? And then at the end here, you have axon terminal, which so these receive the impulses, these take the impulses and take it to the next cells, right? Okay. Now, depending on your dendrites and your axon terminal, you can have different kinds of neurons. Multipolar, both side dendrites, unipolar, only one side dendrites, bipolar, you also have a receptor here and a here, and pseudo unipolar, which basically the cell body is somewhere in between, not following the canonical structure, right? Now, coming to this synapse, which is a very important feature in the transmission of information for neurons. So you can see here, right? You have this channel here. This is called presynaptic channel. This is called prosynaptic. Synapse is basically a gap. This is one of the this is the end of one of the neurons. This is the start of the other neuron. This is the dendrite. This is the axon hillock or axon terminal, right? You get the information, the neurons get the information and then transmits it to the next neuron. And that happens via the synapse. And it uses various neurotransmitter to transmit the information. Okay. So I tried to keep it very simple. So this is basically via synapse. Okay. And it's basically a junction between two neurons. You have presynaptic terminal, pre meaning the one that is bringing the information. Synaptic cleft being the gap, and postsynaptic being the postsynaptic membrane being the member or being the neuron that is receiving the information after the gap. That's why pre and post. Okay. Now, presynaptic terminal has axon terminal of the sending neurons, and you have the synaptic vesicles. Okay, these vesicles have neurotransmitters. Okay, I will go to the neurotransmitters in a while. Then you have this cleft, and in the postsynaptic membrane, you have the receptors for those transmitters which got this information. So you have neurotransmitters here, right? They have receptors for those. So they take the information from the preceding neuron. Okay. Now the synapse are, like I said, is chemical because of neurotransmitter, and there is also electrical synapse, which is direct because of gap junction. Remember, in plants also we have direct connections, right? So now let's try to understand what happens in neurotransmission. How does this transmission of information takes place? So you have these neurons, which basically are bringing the information. The whole impulse generation in the neuron started with an electrical signal. It is converted into a chemical signal and then again into an electrical signal. So both the signals work together when we look at the transmission. Okay, so let's try to see that. First thing foremost, in a neurotransmission, you begin with the arrival of action potential. Not going into detail because that is not of that is supposed not, like, if I dedicate time to this, then we'll not be able to discuss other parts. But you, if you have any doubts, you can refer to, like the action potential and how it is generated and what is the integrities of that. But in broad terms, you have an electrical impulse which reaches the axon terminal, right? You have this conduction of information and it reaches the axon terminal. As soon as this reaches the axon terminal, which is here at the end, okay, the first thing that will happen is calcium channel opening. Okay, so as soon as you have this impulse coming, you have a voltage gated channels here. A lot of these, as you can see here, these ones you see. So these are voltage gated, means they are sensing the change in concentration and ion concentration, all of this, right? So as soon as they receive the impulse, they open up. As soon as they open up, calcium enter inside the cell. Okay. Now, as calcium enters, it triggers synaptic vesicles to fuse with the presynaptic membrane. So these vesicles you see, as soon as calcium comes, these leads to the formation of a bond, a closure between these vesicles and this lining. So this lining and these vesicles fuse. Okay, so that happened because of calcium. Next is neurotransmitter release. As soon as these presynaptic membrane fuse with the presynaptic vesicle, they open up, and the whatever is the content of the neurotransmitters inside the vesicles is released into the synaptic cleft. And then you have the receptor on the other hand of the next neuron, which senses those neurotransmitters, binds to them, and then what happens is, once they are bound, inside these dendrites, which is basically inside those cells, you have other impulses, next action potential generation, and the information is taken forward for that cell. So that's neurotransmission. And here, different neurotransmitters elicit different kind of responses, right? For example, acetylcholine. What is acetylcholine doing? Acetylcholine is coming into any action which is to be done with muscle contraction. You sense something heat, you take your hands back. That taking your hand banks has to be very quick, right? And that happens because of nerve impulses. It goes to the muscle and lead muscles to contract, right? And which neurotransmitter gives that information? Acetylcholine. You also have dopamine, which is for your motor control, right? You have serotonin. We get happy when there is more serotonin. So that's mood regulation. You have glutamate, which is excitatory. So you basically, excitatory in the sense, your nerve cells get excited, means they carry impulse, right? They propagate the information further. So that's glutamate. But you also have GABA, which will do the opposite. When you want the information to be stopped, you have GABA. So your neurotransmitter determine what kind of information goes, if it goes at all or not. Okay. So basically, neurotransmitters or neurons are very important for conduction of signals. And if they get problem, if there is any deficiency or problem with any of these components, you have several problems. For example, dopamine problems, neurotransmitter problems can lead to Alzheimer's or Parkinson's or depression. So all of these, right? So they're all interrelated. Okay. And the last thing that I wanted to discuss here was the types of neurons, right? I told you the type of neuron based on their structure. I will also tell you based on their function. So we have sensory neurons, we have motor neurons, and we have interneurons. Sensory are the ones that carry signals from the receptors to the CNS. They sense it. And the ones that bring the information back are the motor neurons. So you sense there is heat. But what should you do to the heat? That information is brought by the uh, motor neurons. Motor means movement, sensory means sensing, right? Okay, I tried to cover as much as I could. Let's hope the questions do justice to whatever different topics that were discussed in the lectures, right? Let's begin. So adaptation potential can be best described as which ion enter the presynaptic terminal to trigger neurotransmitter release. This we have discussed. Which neurotransmitter is the primary excitatory neurotransmitter in the brain? And self-avoidance in neuron prevents what? So we'll discuss what is self-avoidance also. Triple CA. Anyone else? Anyone? It's easy, right? Neurons and these concepts. These are for your revision because like the ending topic of this course is specialized cell type. We have done all the processes, all different methods in cells. The last part is just to, it seems very repetitive, but it's just trying to bring all of those concepts together for the cells that we already know of, right? Okay, so B, C, D, and C. Let me try to explain to you. Action potential is basically temporary reversal of membrane potential. If I hope I didn't get time to explain that. Action potential is when you have the change in the sodium and the potassium channel, potassium ions across the cell membrane, right? So when there is a change in these concentrations, you have these impulse, opening of vesicles and extrusion of ions and intrusion of ions to maintain that stability. So in general, in homeostasis, you have a certain concentration outside and inside of both the ions. Now, when your concentration reverses, this is what it is referring to as temporary reversal. When it reverses, you have this impulse generation that there is an information that has to be taken forward. That can be because of any voltage gated receptor, any cue, and the action is or the potential is propagated so that this can be reversed and brought back to normal condition. I hope that explains. But uh, the idea here is that uh, to begin information or to send out information, there is this change that has to be done in the initial step, right? Okay. So which ion enters calcium? This we discussed already. Which neurotransmitter is excitatory? Glutamate. GABA I told as the inhibitory. And self-avoidance is to prevent synapses with the same neuron. Now, see, the idea is that I propagate the information to the next neuron so that it reaches to the target faster. If I make a synapse with my own self, it's basically a useless synapse because the information comes back to me. I don't need I already have that information. I need to give it to the next partner. So generally, neuron cells try to prevent this self synapse, formation. Okay, that's to relay the information as efficiently as possible. Okay. All right. The next one was, yeah, so the next set of questions, which neurotransmitter is commonly associated with muscle contraction? I had mentioned this. Which of this is not a part of neurons? What is the primary function of dendrites in neurons? And the long projection that carries nerves impulses away from the body is called. So this is part of the structure of the neurons. TCBB acetylcholine. Okay, one more response. You can see, and then we discuss the answers. CNC, anyone else? Okay. CCBB. Most of you are writing PCBB. I'm seeing someone typing, so I'll wait for a minute. Okay. So I'll give the response. C and C, B and B, most of you wrote that, and it was correct. We already discussed about role of acetylcholine in muscle contraction. Which of this is not a part of neuron? Sarcomere, because that's part of muscle. Axon, dendrites, and soma are part of the neural structure. Primary function of dendrites is to receive the information and process the incoming signal and then transmit it across this body. And that body is axon, right? Okay, and the last set of questions for this topic. Microtubules and axons are oriented such that there's what? Which proteins stabilize microtubules and axons? Which supporting cells produce myelin in peripheral? This we discussed. And neurons, this also we know. Neurons in adult cells are usually found in which cell cycle stage? Any? Kindly share. Sorry. Yeah. Kindly share this PPT at the end of session. So the link I shared will have this PPT also. It already has a 7 other PPTs. It will also have this 8 PPT. I'll upload just after the session gets over. Okay, PBCD, last one G naught. Anyone else? Any other response? Okay, so let's look at the answers just a second. Hmm. So the answer is B, B, C, and D. I believe that's what you have written. The C and D, and E, sorry, that's 4, 2. I don't know why on the slide it shows 2. Just a second, let me correct it. Yeah. Yeah. So the fourth one we already know that neurons are one of the cells that enters permanent cell cycle as G naught. And Schwann cells are part of the myelin, which form the myelin for the peripheral nervous system, oligodendrocytes for the central nervous system, right? Now coming to this, microtubules and exons are oriented such that their plus ends points toward the exon tip, and the tau protein stabilizes these exons, microtubules on the exons. I will just quickly tell you about what microtubules and exons are referring to and what are these plus and minus. So the microtubules that we know already from the previous discussion, in exons they have uniform polarity, right? Sorry. So you have a minus end. Minus means facing towards the cell body. And so I will take you back to the slide maybe. Yeah. So the minus microtubules, minus end of the microtubule will be towards the cell body, which will be this side. Okay, inside the facing this side. And the plus end will be towards the exon terminal, which will be facing this side. Now, why is this polarity important? Why is there this uniform polarity? This ensures that there is direct transport of the material. So I told you, right? The propagation is from this side to this side, and the microtubules is facing, the extension of the microtubules is facing towards this side to propagate those information. Okay. And what are the, so I will, there are two molecules which are most important for microtubules of exons. So you have your kinesin and dynein. Okay, I will not go into much detail. So if you have movement from soma to exon terminal, the motor protein uses kinesin. Okay, and you basically carry the synaptic vesicles, you carry mitochondria, everything else. But if you have a transport in the opposite direction, which is from terminal to soma, that can also happen for growth factors, damaged organelles, all of these. Those come from dynein. So there is this protein, uh, like the orientation of this protein is very important. And that's why this polarity matters in the cell, because that will determine how is dynein arranged and how is kinesin arranged to ensure that the information flows in the correct direction. And tau is one of the protein that is associated to bind to the microtubule and make it stable. Okay. All right. So yeah, let's move to the last topic of this course of this today's session of the week 8, and we'll end it up after that, right? So cancer is the last specialized type of cells that we will study, and we have been doing questions on cancer in the previous session also, that we have a fair understanding of what cancer cells are and what are their features. So let me just walk you through once again. So again, cancer is a disease which is characterized by loss of normal control of cell division. When you have abnormal cell division, you have this cancer state. And you basically, these cancer cells, as you can see on the slides also, they have several key characteristics. For example, unstrained cell proliferation, yes, loss of growth regulation. They have the ability to invade surrounding tissues and also to metastasize. So basically they can move to distant organs, right? So see here, this one here compares and contrasts the healthy and the cancerous cell. In healthy, look at their cells. There's good larger cytoplasm. They have a spherical shape, a nucleolus, and if you look into the genes of the cells, you will have that the tumor suppressor genes are intact and the apoptosis genes are intact. But when you go to cancer cells, look at the morphology. The morphology only tells that this problem with the cell. You have a smaller cytoplasm because they have shrinked. You have a non-spherical shape. It doesn't matter for the shape of the cell to stay, accurate anymore. Multiple nuclei because you have multiple cell divisions, and you have, when you look into the genetics, you will see the tumor suppressor gene and the apoptotic genes or the apoptotic genes are all unregulated and they have all misfunction, right? Malfunction. Now, in general, when you have these cancerous cells accumulation at one place, it's called tumor. Tumor, if you remember, there are two types. There are benign and malignant tumors. Benign are the one that localize and stay where they are, so they don't like they move to the distant organ, they are not invasive. Malignant tumors are the ones which invade and they are spreading via the blood and the lymphatic system. So that's why angiogenesis is very important feature of cancerous cells, right? Okay. And then you have basically, like I said, if you look into the genomics of these, or if you look into the genetics of these cells, you will see that there is not only the cells, the genes have, for example, the tumor suppressor genes, they have lost their function, they have become defective. The proto-oncogenes have gained mutation, they have become functioning, so they will start amplifying and translocating all of this. You will also see that there will be genomic instability. You will see chromosomal abnormality in these kind of cells, as you can see here also. You can see a point mutation, you can see a translocation. They can just basically cut paste anywhere and go, and these are proto-oncogenes, right? So they can mutate, they will become oncogene. They will translocate to another region and start get under a promoter and start expressing. So all of these can happen, and because these happen is why you see cancer. So this genomic instability is definitely there, and cancer. So a lot of genes are classified as tumor suppressor, apoptotic, proto-oncogene. All those cells or all those proteins are, or focus of cancer research, right? There are some other features that cancer cells acquire. For example, they have abnormal shape and motility. We have already seen that. There is loss of contact inhibition. In general, if a cell comes in contact with another cell, it stops proliferating, it stops growing. It's called contact inhibition. You contact, you stop. But in cancer cell, that doesn't matter. That's why they can form these lumps, because there is no, like stop signal if you encounter another cell in the vicinity, right? And there is encouraged independent growth. You don't, these cells can basically grow on their own without needing any external support, right? So they don't rely on any other factors as well, right? And the other thing is their metabolism. So these we discussed last time, which is called Warburg effect. So you see cancer cells with increased glucose intake because they have to multiply and divide faster. And that's why instead of oxidative phosphorylation, which is the normal cell cycle of cells, they do aerobic glycolysis. Even in the presence of oxygen, they can go ahead with glycolysis, right? And they produce large amount of lactate so that they can see, the idea is here is to proliferate. The idea here is to not to maintain the cell integrity. So they just divide, right? And like I said, there are some genes which are classified here, some are here. So proto-oncogenes are RAS, MYC, these kind of proteins which are very commonly known. Tumor suppressor, you have retinoblastoma, RB, P53, BRCA1, BRCA2, these are for breast cancers. And when does a cell finally become cancerous? There are hallmarks of cancer, just like hallmark of cell aging. You have hallmarks of cancer. You have sustained proliferation, resistance to cell death, no apoptotic genes functioning, replicative immortality. So you are immortal, you keep multiplying, you are basically there forever. Angiogenesis, as I mentioned, invasion and metastasis. If you are a malignant tumor, then you have also the capability of invading and metastasizing. And immune evasion, because you are not detected by immune system. They are sometimes detected at the very late stage, so they can evade immune system also. Okay. Right. That brings us to the last set of 12 questions for this course, and then we can finish the lecture today. Right. So which are the two defining characteristics of cancer cells? A tumor that remains localized, does not invade, is called which type of tumor? Spread of cancer cells to distant part is called what? And what type of cancer arises from epithelial cells? So the type of cells also, so the name of the cancer comes from the type of cell that it is originated from. That's the question about. Easy, right? These one and these at least these are not difficult on the slide here. BCBB. Okay, anyone else? Okay, anyone else? Any answer that you want to attempt? Okay, let me give the answer so that we can end this class soon. So what are the two defining characteristics of cancer cell? Uncontrolled proliferation and invasiveness. That's because they lost the normal cell cycle regulation, right? Uh, Tumor that stay at one localized in one region and do not invade are the benign tumors. The one that do are called malignant tumors. And the property of that is called metastasis. Metastasis is basically they can travel from one region to another via blood. And cancer cells or cancer that has arisen from epithelial cell is called carcinoma. The other sarcoma, leukemia is blood, lymphoma is lymph cells. So you can see that the name originate from the cell. You can trace down to the nature of your cancer. That's how we treat it also, right? We can know it's pancreatic cancer because it's come from pancreas. It's localized in pancreas. And then you can also go one step below and see which type of cell has led to the aberration. Okay. Next, most cancer cell originate from which of these options? Which metabolic phenomena is this we have discussed already is observed in cancer cells? Loss of contact inhibition in cancer cells leads to what? And which enzyme helps cancer cell maintain telomere length and avoid replicative senescence? This is something that was connected with the telomere discussion that we had about cell cycle. How does cell age? Telomeres. How do if cell age just stop dividing? Why don't cancer cell do the same? What is the maintenance feature or what is maintaining it? Triple CB. Okay. Anyone else? Any other response? Okay, let me give you the answer. So most cancer originate from a single abnormal cell. Cancer cells are mostly clonal. So one of the cell become aberrant, get a mutation, and then keeps replicating and increase the mutant load. Okay. Yeah, which phenomena is commonly observed in cancer cells? We discussed this already. Warburg effect. Loss of contact inhibition in cancer cells leads to cell piling on top of each other because they are not stopping at all. Even after contacting another cell, and which enzyme help cancer cell maintain telomere length? Telomerase. So cancer cells activate telomerase, and This telomerase activation, because it repairs and maintains the length of telomere, allows them to, bypass Hayflick limit and divide continuously. What is Hayflick limit? Hayflick limit is the maximum number of times a somatic cell can divide before aging, right? And you pass that cap. You don't, you are not restrained by that fact anymore, right? Okay, and the last set of questions. Which gene class become cancer-causing genes? Basically, gain of mutation function leads to which set of these genes become like cancer-causing? Which tumor suppressor protein blocks cell cycle progression by binding E2F transcription factors? Activation of which pathway commonly stimulates cyclin D expression in response to growth factors? And which tumor suppressor protein induces cell cycle arrest in response to DNA damage? Okay, any other response? BBCB, okay. Anyone else? Any response, any question that you want to attempt? I'll wait a minute more and then we can wrap up this session. Okay, let's move ahead. Okay, so this we already know, right? Proto-oncogenes, they are proto-oncogenes. It means they can become oncogenic. They are, proto means they are more favorable to become oncogene. That's why gain of function gives them that feature, right? Which tumor suppressor blocks protein blocks cell cycle progression by E2F transcription factor? Retinoblastoma, right? So what does retinoblastoma do? It binds to these transcription factors, prevents transcription of genes required for S phase entry. Okay, and then what is it doing? It's basically stopping the cell replication. Retinoblastoma is impaired in cancer because if retinoblastoma is functioning, the cell will not undergo S phase or M phase. Cancer cells want to progress to cell cycle phase, so these will be impaired, right? Similarly here, that's why tumor suppressor protein goes defective. Next one says activation of which pathway commonly stimulates cyclin D expression? RAS, RAF, all these signaling pathway, right? Why? Why are we talking about cyclin D suddenly here? Because we are talking about cell cycle. Cell cycle is what leads to replication, and all of these signaling pathway are upregulated in cancerous. They are cancerous cells. They're continuously working, right? And the last one says, again, which tumor suppressor protein induces cell cycle arrest or apoptosis in DNA damage? P53. This P53, which otherwise also called the garden of the genome, is also defected in the cancers because it also doesn't function. So retinoblastoma and P53 are tumor suppressor genes. Obviously, they have to get defective. Otherwise, cell in normal condition, they are the one who rectify the damages to the cells, right? Okay, that brings an end to this session. I will just quickly walk over with you of what we did in this course and then we wrap up. So remember, we have at 8 weeks of this course, there are 8 problem solving sessions. All of these videos and PPTs are available. YouTube videos for the sessions. And I shared a link also. Otherwise, also link is shared with you where you have all the PPTs, right? So we basically started with understanding what is the cell structure, what is their biochemistry, components of the cell, how are the cell information, very general cell processes, processes that maintain the cell homeostasis. Then we began with cell cycle and the regulation. What are the different phases of the cell cycle and how they are regulated? We did about ubiquitin proteasome system, which is one of the important system for apoptosis and cell death. So if we are knowing how the cell are thriving, we also wanted to see how cell are getting killed. We then move to each steps of stages of cell cycle: S phase, G1, G2, M phase. S phase we did in this detail, so at DNA replication, where we also discussed about the centrosomes. Then we after discussing these centrosomes, because they are also duplicated in DNA replication stage. Then we move to the M phase. We looked at mitosis in general. We also looked at meiosis in great detail, different stages of meiosis, 1 and 2. Then after mitosis and meiosis, how do the two cells form? That's because of cytokinesis, right? Since we already did proteasome system once, we again went back to cell death. How does cell death happen? And we looked at what are the natural processes of cell death, like how does it reach to the age of cell death and how does cell division stop? We looked at senescence and we looked at aging, right? After this, we moved to how the cell structure is maintained. So we looked at nuclear organization, chromatin organization, and the three proteins which are responsible for keeping this integrity: SMC proteins, condensin, cohesin, in great details, how they are organized, what they are organized for, and what is their role, and in which step they are functioning. And the last thing that we did is, after understanding the cell biology in general or cellular structures in general, we moved to diversified cells. For example, we started with budding yeast, which we did last session, and today we did plant, neuron, cancer, and stem cells, right? So it gave you a holistic understanding of cell processes and cell biology. And yeah, the topic is very easy. And the course is not difficult. There are huge number of glossaries and huge number of concepts to remember, but they all have logical flow. So all the best to anyone who's taking exams, and hopefully you have gained some knowledge or some understanding of the course from these sessions. And always welcome for your suggestions. If you have any suggestion for me, please put in the chat box. Else we end here. We stop these sessions here today. Thank you very much. I wish you all the very